Okay, so nice to see you. This is my first time to join this community, so probably I only know Professor Aonami Sensei and Kyoda-san in this room, but today I'm going to share our experience in the Database Center for Life Science, where we put a lot of effort to integrate the life science databases. So the mission of our center is to utilize those heterogeneous data sets. And what we achieved so far is including standardization and interoperability of the databases. And to make it possible, we have been organizing annual biohackathons since 2008. And there are some next challenges, and probably images or unstructured data. The usage of those things are most important part, I guess. So the integrating data is crucial for understanding the secrets of life. So for example, in this review article, very famous Daniel MacArthur says that our primary challenge in next decade will be how to expose the mystery, the mechanism, by molecular mechanisms behind the diseases, so molecular interactions or what kind of molecular networks makes the disease happens to humans. So to make it possible, we need to integrate all the data like gene expressions or protein structures or drug protein interactions or glycan modifications. So that is one of the idea of the goal of our data integration. And last year, I finally sequenced my own genome and by out of my pocket. And this is a variant which determine the earwax type, dry or wet, and my type was dry, not so rare in Asian countries. But to reveal the meaning of my phenotype from the genome sequence is still very, very difficult. So we need to look at many databases. So for example, variant databases and clinical databases and protein structure database. So to visit those different databases takes a lot of time, and which database to consult itself is very difficult for beginners maybe, and how to navigate those databases. And each database has very, very complex data structure, so where to see is also a very, very difficult question. So on the first stage, we developed Togo ID conversion service. And in here, we accumulate about 100 databases, and there are networks of the identifiers of the database. And if you have a NCBI gene ID, but your web service doesn't accept that, and only ensemble gene ID is accepted, then user need to convert that before applying the analysis. And also, by using the ID conversion, you can navigate from variant identifier to genes, and the genes to transcripts, and the transcripts to proteins and the structures. So we often face the need of the ID conversions um, since the beginning of the bioinformatics. So this is the latest version of the ID conversion service we are providing. And you can use the very well designed web interface, and the same functionality is also available through web APIs. So you can make it make use of this API through your programming language. And we also defined ontologies between the identifier conversions. So glycans to protein means this protein attach, this glycan attached to this protein, or this protein produces this glycan, or such kind of annotations are also added. However, so this is enough usable, but to utilize the database content itself, we also needed to standardize the data format and the data contents. So that is why we introduced to use semantic web technologies, also known as knowledge graphs or linked open data. So this is a, probably some of you already know about the semantic web or knowledge graph, but it is very, very simple data format, like a subject and a predicate and object to define all the data. So you can easily integrate heterogeneous data set in the same format, and you can query the subgraph of your interest by writing a Spark query like this. Looks it's similar to SQL for RDB. So, and Spark query itself is a web API, so it is open to the world. So that is findable and accessible from the world. And interoperable means you can integrate heterogeneous data sets, and you can also reuse the data someone put into the graph of the network. So we choose to use this infrastructure in 2010, but still international collaboration was needed. What kind of data models we use for genes or phenotypes, and what kind of ontologies we reuse, or do we need to create something new to represent this new ideas? And we should have a globally unique identifiers like URIs, and but which URI to use, and what kind of metrics, fair metrics, you maybe evaluate, and workflows and the reuse of modules. And so to make it community agreement, we have been organizing biohackathons since 2008. And so far we have organized 14 times, and that was a great opportunity to discussing with the database developers like a Uniplot database or EBL guys, so we can harmonize what can be standardized. And so far, we have been attracting 100 participants every year, and 30% of the participants are invited from overseas, and another half are from from Japan. And it's a one-week event, and on the first day, we're setting up the hacking topics for the week, and we have basically a free time to code or discuss or do anything. And on the last day, we wrapping up summary document, which can be submitted to our own preprint server, biohackathons, because those 1 week summary of the hacking event was not accepted to the bioarchive, so we needed to have our own one. Okay, anyways, we have a great international community like this, and we created a lot of open source software and ontologies and principles, and actually one of the fair principles, idea of the fair principles are incubated in the biohackathon in 2015. So as a result, we could develop RDF datasets, knowledge graphs, on the most of the bioinformatics major domains of the bioinformatics, like from genome sequences to proteins to drugs and glycogen proteomics pathways. So, and all those data sets are stored in our RDF portal. So where you can use the Spark endpoint to extract information from each databases. So, but still, the learning curve to utilize those information is still very, very difficult. So we have been developing a lot of supporting tools. But for example, in INSDC dataset, we have 3 billion sequences, and each original dataset looks like this Fortran style text format, but we pass it and chopped it and made it into subject and predicate object. So in total, we have 10 billion lines of those. And in Uniprot, they also have 300 million proteins, and which is represented in probably this one was 5 billions of triples. I mistyped. So, and the data structure of the Uniprot is very complex, and for protein structure database, PDB is also converted to RDB by PDBJ in Japan. So they only have 200,000 records, and those information is already stored in a knowledge graphs, but by using those information, the structure prediction service like AlphaFold is also developed. So having a um, reference information in those databases are very important. And PDB is itself is also very, very complex. So where to find how many oligomers this protein forms or something like that is somewhat difficult. So probably you need expert knowledge and to identify where on the web page or where in the graph structure to find your data. And by knowing that, you can finally start writing a code to extract the information. So maybe we should combine the ChatGPT UI or something like that to make it automated. But so far, for now, you need to understand what kind of information is stored in this database and in which data structure. So anyway, so we have a collection of those RDF datasets converted from the original data formats. And so we can now use Spark or common language to query the graph database. And we have been developing a lot of middleware to make it easier. And as finally, you can visualize it or analyze those data. And because the title given to me was a tools for data integration, so let me briefly introduce several tools we have been developing. So one of the tools is Sparkle proxy, which protects your Sparkle endpoint and which also caches the Sparkle query results. So it can be utilized in a high performance situation. And Spark list. So probably you don't want to write a long lines of Spark query again and again. So you can just make it a markdown document, and then you can just call the REST API by assigning parameters to execute the same Spark query, only changing the parameters. And RDConfig is another type of tool to describe the very, very complex graph structure as a free structure. So for humans, actually, the graph structure is very difficult to interpret. So if you cut some links between the graph nodes, then you can treat main passes to the information from starting from one protein and a lot of attributes like a tree structure. So by preparing the data model in a YAML data format, then you can automatically generate Sparkle queries, or you can create beautiful schema diagrams, or even you can make a GraphQL schemas. So here is a command line to generate a Spark query. So if you are only interested in names of Japanese sake and the category of the sake is Junmai, then this Spark query is for you to extract your favorite Japanese sake. Okay, so the final tool I will introduce today is a Grasp. So this one convert your Sparkle endpoint into GraphQL endpoint. To configure it, you need to write a mapping between GraphQL's query to Sparkle query, but RDF config can automatically generate it. So here is a still very small, but here is a query for Uniprot by assigning one identifier, and you can easily extract the information like a protein annotation or protein sequence in a JSON. So this will be much easier for web developers and database users. So we are now trying to utilize all those infrastructures. And going back to the data on the life science data, we firstly started to create an integrated genome database by only using RDFs. And we integrate a lot of different types of the biological attributes. And we successfully made up to host 20,000 of species in Togo genome database. And by taking out human subset, we created a Togoba database. This is integrated Japanese genomic variation data. So by taking the way to utilize a graph structure, it could be get much easier to integrate heterogeneous data. So first learning curve was very steep, but after utilize, get used to the technology, then we can easily integrate any types of the data in the end. So here is a gene report and a variant report and disease report, and each of those information are taken from the different data sets, but all those data are stored in a single Spark endpoint. And interestingly, you can reuse all those visualization components by using the Togo standard technology. So this is a generic web component, so you can just copy and paste those 1, 2 lines of the HTML in your web page. Then you will have this genome browser or those variant frequencies in your database. So maybe, and this technology is very generic, so maybe you can develop your own database by using those concepts. So you can reuse the same information, and you can reuse it in related databases. So someone need to develop once, but you can reuse it in anywhere. And how to show the power of database integration was the last question to us, and funding agencies are always angry about that. So do you stop to integrate, or what is the outcomes of the data integration? So, and it is actually a responsibility of the scientist, but probably we needed to show it in some proof of concept application. So first one was this Togo DX Data Explorer, in which we integrated those information related to humans. And you can see the bird's eye view of the distribution of each gene or each diseases, and all the links between those genes and the proteins and the pathways and diseases and variants are using Togo ID conversion tools. So you can put any identifier of the supported database, then you can traverse to another layer of the information in one view. So here are some examples of the attributes for genes and the proteins structures. So you can choose any of those attributes to narrow down. No, this one figure is showing the enrichment analysis. So you can put any identifier like a gene symbol or how to say PubChem identifier for chemical compounds, then you will see where your identifiers are biased, enriched in the entire view. And you can also use this interface for the exploratory data analysis. So you can specify like genes expressed in lung or intestine and are localized on the cell surface and having a 3D structure in PDB to find candidate genes for drug targets of the COVID-19. So such kind of the very complex query can be automatically executed in this system. But this was not so catchy especially for old people, and even for me, it's too small. So recently we created a new visualization exhibition. So you can, we use the same data like gene expression and of the, so how to say, gene expression and disease-related genes are plotted here. So, and as a gimmick, here is a motion sensor. So if you use a stand in front of the screen, this organ human is walking like you. And the genes related to, for example, heart disease is mapped onto the organs, and yeah, and the protein structure of those proteins are shown here, and you can also map the pathogenic variants on the figure of the protein structure. In the bottom here is a evolutionary scale. So how many genes? For example, in this specific heart disease, we had 11 genes, but when in evolutionary history those genes are acquired in human, you can see the orthologous gene record to see. And interestingly, most genes are acquired in the vertebrates, and one gene is only appeared in chimpanzees and humans. So this is just giving inspiration for new hypothesis. But I think looking at the same data, but in different ways, is probably important thing. So I would like to make it available at the scientific museums. So, yeah, that's it. Thank you.